Hi, this is Brad Power, and this is the Cancer Patient Lab, and we're honored to have today with us Ray Kurzrock. I've known Ray for many years, first from working with CureMatch, which helps people identify multiple drug combinations based on their biomarkers, and worked with Ali Perlina, who's their chief science officer there for many years. And then also she's with the Target Cancer Foundation. She's the force behind it, which helps people with rare cancers get um, both genetic analysis from a blood test as well as gets a virtual molecular tumor board to review their case and make recommendations. And she's had clinical trials that have supported both of those activities. So she's at that intersection of being a pioneer and also gathering evidence that supports her pioneering work. So we're very honored to have Ray with us today.
So thank you for inviting me, and it's a pleasure to talk. And I'm going to give a talk of about 15 minutes, going over where I think cancer therapy needs to go. And I think we need to change what we're doing if we want a different result. So we need to move from being drug-centric to patient-centric. And this is an evolution. Also moving from looking at cancer by organ of origin as a colorectal or breast to tumor agnostic to N of one, understanding that every patient is different and therapy needs to be customized to every patient. And so one of our biggest problems, I think, is us. So Edmund Hillary, who was the first person to climb Mount Everest, said, "It's not the mountain we conquer, but ourselves." And more and more, I'm finding that's true for cancer therapy. Cancer therapy needs to change, but the standard treatments are very entrenched and it's very difficult to change, even where the treatments are not very effective. So these are my disclosures. And let's go over what cancer is. Cancer is basically caused by genomic DNA errors. DNA is the code for life, and there's errors in the DNA that causes cancer in cells that the immune system fails to eradicate. So it's about the pillars of precision personalized medicine are genomics and immunotherapy, because both systems have failed. The DNA errors, the DNA has created errors, and the immune system, whose job it is to get rid of cancer cells and infections, has failed. Mainly, it's failed because the cancer cells, in order to survive, have put the immune system to sleep. So our basic premise is that every metastatic tumor is distinct and complex. The analogy, I'm from Canada, so malignant snowflakes. Every snowflake is has a crystal structure that is very complicated, and every snowflake is different from every other snowflake. And as we learn more about cancer, this is clear that this is true for cancers too. And therefore, in order for treatment to be precise, which is what precision medicine and what my whole orientation is about, it must be personalized. So, to be precise, everybody gets a different treatment that is customized for their cancer. Um, so one of the take-home points is patients, not drugs or clinical trials, should be the center of the universe. And so what we have historically done is, especially in the clinical trials realm, we have a clinical trial and it's testing drug A or drug A and B, and then we fit patients onto that clinical trial. But what we really need to be doing, and what we've tried to do in our recent I Predict trials, is we have a patient at the center of the universe, and we fit the drugs to the patient, not the patients to the drugs. And just a little bit about who I am. So I'm a professor of medicine and director for the Center for precision oncology and rare cancers at Medical College of Wisconsin. I'm also the Chief Medical Officer for the Worldwide Innovative Network for Precision Medicine, which is a global network. And just a little personal, my husband's a physician. I have 4 children and 3 dogs, which are also our children. And I like to travel. So we were recently in Greenland in August, and it's absolutely beautiful. And this is the setting sun. I understand it might become our 51st state, but we'll see. Okay, that's a joke. Um, okay, so my experience, I did my fellowship training at MD Anderson Cancer Center. I said I came from Canada, and I grew through the ranks. I became a department chair. I founded a new department for early phase clinical trials, which grew to be the largest of its type in the world. And we instituted, as part of the fabric of the department, um, uh, precision medicine, molecular typing. And we were involved with early phase trials for 8 drugs that are very familiar to you that are now FDA approved. I moved on to University of California, San Diego, and I was founder and director for the Center for Personalized Cancer Therapy. I've overseen molecular profiling on more than 21,000 patients. I was very honored to be part of the White House Cancer Moonshot initiative. I'm the vertically compromised person beside Biden there. And so very involved in trying to move the field forward. So I think there's a new era that has been born, and that's what I alluded to at the beginning, and that's the genomics and the immunotherapy era. And so let me go through what we have done for most of my professional career for cancer and what we're still doing, but why I think it's not the right thing for patients. So the traditional method of deciding therapy based on light microscope diagnosis. So the first thing we do is we diagnose patients. Do you have colorectal cancer? Do you have lung cancer? Do you have breast cancer? And then the diagnosis, we provide therapy. But essentially, by doing it this way, we are guessing what the therapy is. And I'm going to try and give you an analogy to make this more clear. So guesswork is figuring out what is wrong with a car by knowing the make of the car. So let's say you have a Ford or you have a Honda. And let's say Fords commonly have spark plug abnormalities. I made that up, but let's say that's true. So if you have a Ford, we're going to go in and we're going to fix your spark plugs. So that, kind of doesn't make a lot of sense because not all Fords have spark plug abnormalities, might have something else wrong with them. And essentially, I think that's what we're doing when we diagnose cancer by organ of origin. You have colorectal cancer, it frequently has this or occasionally has this, so we're going to give you this therapy. That's really guesswork. So if you want to fix your car, you have to look under the hood to get an accurate diagnosis. I don't care if 50% of Fords have spark plug abnormalities. You really want to know what's wrong with the Ford in front of you. And the only way to know that is to look under the hood. Okay, so let's go back to cancer, where this analogy comes from. The way we diagnose cancer is from the light microscope, which was invented in 1590. So we've been doing this for a long time. And it's really a great instrument, but after 400 years, it may be time to move on. And what this tells us is what's on the surface of the cell. It tells us, oh, that cell looks like it came from the colon, so it's a colon cancer. That cell looks like it came from the breast. It's a breast cancer. Doesn't tell us anything about what is wrong under the hood. So figuring out what is wrong with the tumor based on the organ it originated from, breast cancer or colorectal cancer, is guesswork. And we need to be looking under the hood to allow an accurate diagnosis. So how do we look under the hood? Well, we have this amazing technology that was actually unimaginable a few years ago. And this is next generation sequencing. So we can actually know the DNA errors in every patient. And beyond that, we can also know now the immune function errors. So the technology has advanced in a way that's absolutely breathtaking. I would say unparalleled in human history. The first human genome was sequenced around 2003, took 13 years and cost almost $3 billion. Today, we can sequence in a day for $1,000. We're actually coming to the $100 genome. So this is now democratized. Everybody can get this sequencing, but it also means that we have to change everything that we've been doing because we've constructed all our thinking around a system that doesn't work well for treating cancers, especially metastatic cancers. And maybe that's why we haven't done so well in treating them. So we created a master protocol called iPredict, which is investigation of profile-related evidence determining individualized cancer therapy, where the idea was that every patient would be treated individually with a personalized combination of therapy based on their biomarkers. So this goes back to what we said. We go from drugs being the center of the universe to patients being the center of the universe. And to my knowledge, this is the first trial that ever did something like that, that didn't put the drugs at the center, but to put the patients at the center. And the underlying premise of iPredict was that cancer in different individuals is, like I said, akin to malignant snowflakes. Each is complex and distinct. And that's exactly what we know from doing genomics on patients. Patients have multiple abnormalities, and they're all different from patient to patient. And we went beyond, I'm going to get in beyond just thinking about individualized drugs, but also individualized dosing. Because in the old days, which is still now, we always thought more drug is better. With chemotherapy, it was clear chemotherapy kills cells, more drug is better. But it's not clear at all that you need more drug is better for immunotherapy or targeted therapies. Beyond a certain dose for an individual, you may be just getting toxicity. So we also wanted to emphasize quality of life, and could we give the right dose for every patient? And this should just make sense. So I pulled these off the internet. These are two different patients that have cancer. I don't think you need to be a physician to realize that these two patients should not be getting the same dose. And what we do is we give one size fits all for all patients. That's not right for these two patients. The 20-year-old robust patient with cancer and the 85-year-old fragile patient should not be getting the same dose. And then this is a real person. What dose does she get? I really like her. But at any rate, so we really need to be thinking about that. So what we did is we did a trial where every patient got a customized combination of therapy that was individually dosed for them. So it was the drugs were customized to them and the dose was customized to them too. And actually, we've published this several papers, which I think I have in my slide set, but the most recent one just came out, I think last Friday in Journal of Clinical Oncology, which is the ASCO journal. And what we show is that there is a linear correlation between the degree of biomarker matching and outcome. It's a very strong correlation with R squares of 0.76 to 0.94. One is 100% correlation. And what we show is that if you match just one biomarker, you get an incremental improvement. But if you match multiple alterations, that is where you really see a tremendous improvement in outcome in response rate, progression-free survival, and overall survival. And so we've published this in several publications over the years. I'm not going to give you all the publications, but just the highlight. First published in Nature Medicine in 2019, Genome Medicine, and we treated newly diagnosed metastatic patients, and we published that. 2021, and then this in Journal of Clinical Oncology, which, as I said, was just published a few days ago. And the most recent publication, um, we had, I think, 157 different regimens in 210 patients. Every patient got their own regimen. Every patient got their own dosing. And what we showed was that the degree of biomarker matching to the tumor corresponded with better outcomes. And we also showed that compromising the dose did not compromise the outcome. In fact, it may have made the outcomes better because if patients can't tolerate their therapy, they don't take it. This mentality of more drug is better that we learned from the chemotherapy era really doesn't apply to immunotherapy or targeted therapy. The right dose is the dose that the patient tolerates well. So this is just a few days ago. The original studies that we had published were featured in National Geographic, The Future of Medicine, in the NIH Director's Blog. I was honored to speak at the Nobel Symposium for Precision Medicine. So it's gotten some traction, but I think it's still very difficult for doctors to change what they've been doing. So I'm going to give you some patient case examples because I want to show you how this works in a practical manner. So this is the first patient I want to talk to about. This is a 39-year-old woman, young woman with adrenocortical cancer metastatic to the liver. So adrenocortical cancer, the treatment doesn't work for it very well. It is almost universally fatal. The median survival is about 9 months. And this patient had what we call Lynch syndrome. She was microsatellite unstable. She had a hereditary alteration in the MLH1 gene, and it's probably why she got cancer at a young age. Now, she got the right treatment, which was Keytruda or pembrolizumab for this MSI high, because MSI high patients tend to respond to immunotherapy. Some of them respond beautifully, but she did not respond to pembrolizumab, unfortunately. And not only that, but she had life-threatening liver toxicity and a huge skin reaction. And so she came to us at our Precision Oncology and Rare Cancer Center. And we discussed, so what are we going to do with her? And what we decided to do was unusual, but that's like our job. So the first thing is patients who fail one immunotherapy sometimes will respond to two immunotherapies put together because maybe the tumor is putting the immune system to sleep so that it can survive, and maybe one immunotherapy isn't enough. So we decided we'd give her two immunotherapies put together. But that's, of course, a problem because the referring physicians say you can never give immunotherapy again. She had life-threatening hepatitis, life-threatening liver dysfunction, which can happen. So we said, you know what, we're just going to lower the dose. We're going to give her a much lower dose. And so we gave her a lower dose. And we added on a targeted therapy, a MEK inhibitor, cobimetinib, because she had a alteration that can be targeted. If she had an alteration in her next generation sequencing, that can be targeted by a MEK inhibitor. And we actually treated her a few years ago, but we've recently published that patients that have failed to respond to one immunotherapy do not have a compromised response rate when the two immunotherapies are given together. So we just published that actually a few days ago. It says in press, but it's now published. So this made sense, but we actually treated her almost 4 years ago. And so what happened with her? This is her tumors in May 2022. Her liver is full of cancer. And then by August, her liver is clean. And this is her actually recently. So we are now, I guess, more than 3.5 years in, and she is in complete remission. She has no liver toxicity. So, what are the lessons learned? First of all, we used her tumor markers to give her a combination of three drugs. That's number one. Number two, and we combined immunotherapy and targeted therapy. And number two, we lowered the dose. And so a patient that had life-threatening liver toxicity from immunotherapy, which mostly physicians will not return to the immunotherapy, has had no toxicity to repeat immunotherapy with two different drugs, but at a lower dose. And she's achieved a complete remission. So, this is the idea of personalizing the drugs and personalizing the dose for the patient. Um, so she's now 3.5 years later with no toxicity, no evidence of disease. Here's another patient. This patient has a poorly differentiated carcinoma of unknown primary. So this is a cancer we don't know where it came from. And often what people try and do is figure out what it came from, where it came from. Did it come from the colon? Did it come from the breast? To me, that's regressive. I don't really care where it came from, as I mentioned. I care what are the molecular alterations in that tumor. So we found that this patient had two molecular alterations that could respond to two targeted drugs. One is trametinib and one is olaparib, which is a PARP inhibitor. And we gave that combination. The combination had not been given previously in phase 1 studies, but what we showed in our recent JCO paper is we don't need previous phase 1 studies. We compromise the dose, and the compromising of the dose is not problematic. It doesn't decrease the response rate, and we get really nice responses. By 16 weeks, the patient looked clear of the disease, and therapy is ongoing at 17 plus months. Okay, so what we're trying to do at my institution is we have an umbrella protocol where we can treat any patient that walks into the cancer center with the next generation of the iPredict study. And we also have created a trial with the Target Cancer Foundation, and Jim Palma leads this foundation, has done an absolutely superb job in operationalizing this trial. And this trial is open to anybody in the country, and it's an iPredict style trial, and they can stay at home. It's completely decentralized, remote consenting, keeps patients at home, and which I think is much better for patients. And then what you can see is we've consented about 400 patients. Patients are from 44 different states. And actually, the people that participate in the molecular tumor board are from 8 different institutions. So we have expertise from across the country, major institutions like MD Anderson, Dana-Farber, Harvard, City of Hope, bringing people together to try and construct the best therapy for any one patient. So next thing I just want to mention is genomics is the tip of the iceberg. So we're doing genomics, but we're now doing RNA, we're doing protein, and we're doing functional analysis and many other things like that. And then we did do a study that we published with the WIN Consortium, which is a global consortium, which was the first trial that incorporated both genomics and transcriptomics. And again, an N of 1 trial published in Nature Medicine, 5 countries brought together through a molecular tumor board and through a single trial to help patients really around the world that are part of the WIN Consortium. So what are we trying to do? We're trying to do discovery in real time and treatment in real time. So in the clinic, as you know, there can be a new discovery, but it can take years until that becomes a clinical trial. But at least for approved drugs under the PREDICT umbrella, we can figure out a treatment within a week and institute it for the patient that's sitting in front of us in the clinic. And molecular tumor board, there's an excellent molecular tumor board. We have an institutional one, one across the country and one across the world. And just give you an example of an international collaboration with a rare cancer that is untreatable. This is a patient with fibrolamellar cancer, a young woman, 32 years old, and she was had multiple different therapies, mostly which don't work for these tumors, and she was told to go to hospice. We had an analysis done by a group called Pangea, which does a AI-based analysis of the RNA and gave us a completely non-intuitive therapy, which happened to be immunotherapy. And so we gave her immunotherapy. And at the time we published her, she had gone from hospice to complete remission, which was 2 years at the time of publication. It's now almost 5 years. So she's probably cured of her disease. So this is bringing all the elements together internationally to help a patient, which is something that I'm very excited about. So I'm just going to finish by looking into the future. The first thing is every patient has to have next generation sequencing. We believe genomics is the diagnosis. We've published this in JAMA Oncology in 2016. We've published it in Journal of Clinical Oncology in 2023. But many patients still do not have access to NGS. But I think this is fundamental. At the time of diagnosis, every patient should get NGS. And then the other thing that we really need to be thinking about is how do we deal with all of this complexity? If there's 300 drugs in oncology, there are 45,000 two-drug combinations and over 4.5 million three-drug combinations. And if there are 700 cancer genes, which there are, and the median number of alterations per tumor is 5, there's over 1 trillion possible molecular patterns. So we need AI-based GPSs that navigate the complexity to optimize match combinations of individualized gene and immune targeted therapies. And I am going to finish, and special thanks to our patients and our precision medicine team. And I think I went a little bit over, but I am happy to answer any questions at this point.
Thank you very much, Ray. That was awesome. There are a couple questions already in the chat. I don't know if you can see them or I can read them to you.
Why don't
Um,
you go ahead and read them to me so that everybody
sure.
can hear them too?
Given the low cost of sequencing and analytical tools now available, can you talk, even if a bit aspirationally, about what needs to happen, regulatory, medical, culture, et cetera, to scale N of 1 treatments more broadly and rapidly?
So that's a really good question. I think that right now, CMS covers stage 3 and 4 cancers that have an indication that's solid tumors. Essentially, with the tumor agnostic therapies, every solid tumor has an indication. So every stage 3 and 4 cancer can get NGS. Our belief, and we're trying to institute it at our cancer center, is that every patient from the time of diagnosis, I don't care if they're stage 1 or 2, and as you know, some stage 1 and 2 cancers don't behave very well either, lung cancer, pancreatic cancer, and so forth. Every patient should be getting NGS. I don't know how to make that happen at a regulatory point of view. I actually think patient advocacy might help, maybe a lot. And I hope that we won't have any setbacks because I think Rec Pazdur was a very strong advocate for everything at the FDA, and I'm not sure that that's continuing, but I think the patient voices need to be heard. On the medical side, there's also resistance, and which is really hard for me to figure out. But I think one of the basic things that needs to be happening is training. Um, we have a precision medicine clinic and we have fellows that come through and they're third year fellows. They're ready to be launched and they can't read an NGS report. This is a total failure of training. These are smart people. They got into medical school. They got through medical school. They got through fellowship, which can be really hard. They just have not received adequate training. I don't.
9 p.m. Union City. Sorry. Um, Follow-up question. You mentioned the FDA. Naomi asks, what is your view of the FDA's recent N of 1 proposal announced in the New England Journal of Medicine last month?
Hmm. I I'm not sure I'm familiar. I think you're talking about the rare diseases and therapy for rare diseases. Um, so I'm not sure we're talking about the same thing. Um, so there was an article about therapy of rare diseases and that we need to move forward with that. Um, so I think if the FDA goes through with that, that would be great. What I am very worried about is um, the insistence that we need a randomized controlled trial for everything. So randomized controlled trials have always been considered the gold standard because they minimize the chance of errors. But they take years and they take a ton of money. And I think the balance sheet is a negative for them when you're talking about lethal diseases. Um, I also think that it's not right that beyond a certain response rate, it becomes unethical to randomize patients to to a control arm. So the better your therapy, the more the ethical question arises um, about doing randomized controlled trials. So I think if the FDA does go ahead with N of one, that would be great. But apart from the New England Journal article, that has not been the position of the people that currently leading the FDA. And so they've been very outspoken about the need for randomized controlled trials, and that concerns me.
Thank you. Glenn Sabin has 3 questions here. How is fundraising going for the support of WIN's innovative WING-PO trial?
We can always use more funds. So the WINGPO trial is something like I predict with the idea that we are going to bring that globally. So WIN has members in probably 18 different countries, every continent, Europe, Canada, the United States, Australia, Asia, Japan. I know some of that's Asia, and China and so forth. And we are trying to do a next generation WINGPO trial, which is N of one, and do it really globally. And we actually have a molecular tumor board now that meets monthly, but it's a global molecular tumor board, which is pretty incredible. People bring patients, and you have experts from 18 different countries that are weighing in and all very innovative. Um, but WINGPO still needs more funding to launch. That's the bottom line.
Next question. Are you using pharmacogenomics to support treatment decision-making and dosing?
We have not used it to support uh, dosing per se, but we all know that. But we do use pharmacogenomics like DPYD if we're going to give 5-FU derivatives. We actually don't give a lot of 5-FU derivatives in our clinic. UGT, if we're going to give topo-1 inhibitors and so forth. The question, so the pharmacogenomics that have come into the news, especially, is DPYD because of the death of Larry Milesky at Dana-Farber, and so I think any patient that is going to get a 5-FU or related drug should be getting DPYD. I think that every patient should get DPYD because you don't want to miss something the way it was missed. And probably it would be missed not just at Dana-Farber, but almost everywhere. And the same is true for UGT. And then the bigger question is, can we use pharmacogenomics to titrate dosing for individual patients? And I think that's a really good question, but people have not done that yet. The other way to titrate dosing, which is something that was introduced to me over a decade ago at MD Anderson, but I don't think anybody has moved forward with it, was there was a foundation that was trying to make a bedside machine where you could take blood from the patient and look at their the concentration of the drug in their blood, just bedside, instantly. And to me, that's just a really good idea. What we've done is in our clinic is when we give drugs together, we've analyzed toxicities, and if we think that there's going to be toxicity, or if the patient is elderly or has other ways they're compromised, we reduce the dose. And we start with a reduced dose. That was against what I was taught and what most people have been taught. But I think what we were taught was wrong. It might have been right for chemotherapy. It's not right for these new drugs. We reduce the dose and then we titrate the dose to the patient's tolerance because we want them to have a good quality of life. We don't want them to come off the drug because they have intolerable side effects. And the concern, which I heard endlessly because it's what I was also taught, is you're reducing the dose. It's not enough drug. These patients are not going to do well. But what we showed, and this is the paper we just published in JCO a few days ago, is that compromising the dose absolutely did not compromise the response rate or the outcome. And my clinical impression is that the outcome is actually better because the patient has a good quality of life and because the patient doesn't come off the drug. So the worst thing you can do to patients, in my experience, is start with a high dose. They have some terrible side effects, and then both the doctor and the patient never want to see that drug again. And then they come to see us and we say, okay, well, how about if we give you a lower dose? And they say, no, I don't want to touch that drug again. And to me, that's a really bad thing. In the patient that I showed you, the 39-year-old with that we gave immunotherapy to, her physician told us, and I understand why he said that. He said, you cannot give her immunotherapy. I'm not going to write an order for it because she had life-threatening hepatitis. You just can't do this. And so we said, fine, we'll do it. And we discussed it with her, and she was kind of worried. But, the alternative was she had no good treatment for her metastatic disease. And here she had a really well-matched treatment. So we had a discussion and we lowered the dose and it went well. So I think we have a lot of different ways to titrate dosing, and we're at the tip of the iceberg of how to do that.
Related to your recently published JCO article, is that available open access?
Yes. We wanted to make sure that everybody would be able to see it. And if you shoot me an email, I'll send you a PDF of it as well.
Thank you. Cindy Ness has her hand raised. Cindy?
Hi, grateful for the presentation, truly. Glenn asked several questions. I thought I was going to have to give some justification for asking several, but now he's paved the way. I'll be quick, though. Of course, it makes so much sense to look at a patient as an N of one. not every patient, even when you're looking at them as an N of one, gets better, survives. So what do you think are some of the greatest hurdles that are still in the way of dealing with patients, even if they're N of one? That was like the first question. And do you see every patient as an N of one having a rare cancer or not really, meaning that there's certain pathophysiology that's understood. And in most cases, that will get you like much of the way. It's only after they fail several kinds of treatment that then you might feel that they're eligible, let's say, to come see you at your clinic. I ask this because I referred a particular patient, pancreatic cancer, metastatic, and for some reason, and I lost track, he was not considered eligible. So I'm just trying to understand who you accept and who you don't accept. And then the last piece, lifestyle management. That's what I do with patients. And I'm wondering, do you have any thoughts about whether nutrition, exercise, stress reductions, things of those sorts really can move the dial on cancer outcome?
Okay, so I've written down your questions, so I hope I have them all. Okay, so barriers to instituting this. So historically, we do a study with the same drugs in 20 patients, and then we tell you an outcome. And now I'm telling you that everybody gets a different regimen. In the JCO paper, we had 210 patients, and I think, don't quote me, I'm pretty close, but I think there were 157 different regimens. Um, so you're going to say, so how do you know what works? Because we're not evaluating an individual regimen in an individual disease. We're evaluating the algorithm that matches them. How good is the algorithm? And I think eventually these algorithms are going to need to be approved. That's what's going to go to FDA approval. How good specific algorithms predict response. And Brad mentioned CureMatch was a company that I co-founded because of this new paradigm. And we have a certain algorithm. And I mentioned Pangea that we used for a patient. They have a completely different, it's scientifically completely different way of predicting. So there's more than one way to skin this cat, and maybe algorithms will come together, but the whole idea is to use an algorithm. Some of the barriers, big barrier is getting the drugs. And we've instituted a medication acquisition specialist. We can get 90% of the drugs we want. But ultimately, this is not scalable across the country. And ultimately, I think there has to be approval for these algorithms so we can mix and match the drugs. Um, the science isn't perfect. Not every target is targetable. Okay, so like RAS was undruggable. Now we have drugs that are coming through. We have P53, which is very poorly druggable. So the science isn't perfect. Um, I think those are some of the barriers, and we've overcome at least the getting drug part of the barrier in our clinic, but this needs to be operationalized and scaled so people can do it anywhere. You asked about rare cancers and common cancer. We consider every patient as an individual, every single patient. We absolutely do not believe you have to have failed treatment. I don't know why the patient didn't get to our clinic, but my pet peeves are actually pancreatic cancer and glioblastoma, both of which have very poor outcomes. And I'm going to get killed by my colleagues for saying this, but I'm still going to say it. I actually think that patients should get anything but standard of care because the outcomes are so poor. With pancreatic cancer, only 20, there was a major study just published, only 24% of patients are alive at 1 year, which are best of standard of care. So it's my belief that you should not get standard of care. That's not a lot of people don't agree with me, but I'm going to say it. Glioblastoma, the treatment we use was from a publication 20 years ago in New England Journal of Medicine. It's now 21 years that showed a 6-week improvement in survival, but everybody's dead at 3 years. So I think patients should get anything but standard of care. but obviously, a lot of physicians don't agree with me. I also think patients should be treated when they're newly diagnosed because they do much better. And yes, we wait to end stage disease. It's kind of almost like a tyranny that you have to give them standard of care no matter how lousy the standard of care is. By the way, not all standard of care is lousy. Some standard of care is curative, but I'm talking for where it is lousy. We have created the iPredict trial so we can treat neoadjuvant, we can treat newly diagnosed patients and so forth, but we don't get a lot of referrals. And I'm going to give you two very quick examples of transformative work by treating early. So the first thing is, before I was a hematologist, before I was a solid tumor doctor, I was a hematologist, and I was very involved with some of the early chronic myelogenous leukemia work. And actually, it'll age me, but I actually published the first P190 BCR-ABL paper in Nature. I was the first author on that. So people don't even know that because they think I'm a solid tumor doctor. But that was when I was very young. I was probably about 5 years old. I'm just joking. But at any rate, so chronic myelogenous leukemia is a model for what can be done. And what everybody says is this so if you we know the molecular alteration in chronic myelogenous leukemia is called BCR-ABL. This is the one that I just said about the Nature paper. And we know what it does. And we know there's a great drug called imatinib that reverses this abnormality. And then, and what imatinib has done for chronic myelogenous leukemia, this disease used to be a death sentence. Everybody died in 3 or 4 years, near 100% death. Now people have a normal, pretty much close to normal life expectancy, and the response rates are close to 100%. So what I hear repeatedly is, oh, but solid tumors are different. They don't respond that well. And what I say is, no, the investigators are different because if you take the drug imatinib and you give it to CML patients in later stages of the disease, if you even wait a year or two from diagnosis, the response rates are about 10%, and the impact on survival is negligible. In order for imatinib to work well, it has to be given at the time of diagnosis. And so we have not learned that important part of the story in the solid tumors. There's another set of studies that I think is hugely important in this space, and that's with immunotherapy. So Andrea Churchick at Sloan Kettering has published 2 New England Journal of Medicine articles, and then there's a group from MD Anderson that published a Nature Communication article on this. So the first New England Journal of Medicine article was she took patients with microsatellite instability and the colorectal rectal cancer, and this microsatellite instability predisposes you to great responses to immunotherapy. But she took these patients and she said, no surgery, no radiation, no chemotherapy. At the time of diagnosis, we are going to give them immunotherapy. And so the first paper in New England Journal was 12 patients. All 12 patients were cured. No need for surgery, radiation, or chemotherapy. She then published a second paper, which was last year. I don't remember the number of patients. I'm going to say like 100 patients. And 92% of them at 2 years are free of disease. 92%. So MSI high predisposes you to response, but in later stages, the response rates are only about 30%, like the cure rate. You still have a cure rate, but it's probably about 30%. And she's getting a 92% cure rate by bringing this up front. And patients don't need surgery, don't need radiation, don't need chemotherapy. Chemotherapy is tough on patients. Radiation is tough. Surgery is mutilating. If you have rectal cancer, you'll often need a colostomy. So I think it's a long way of saying I absolutely believe we need to bring this up front. And our study allows us to do it, but it is really hard to get the referrals. People do not want to circumvent the standard of care. So that's, I hope I answered your questions. Oh, nutrition and exercise. So yes, of course, I think nutrition and exercise is important. Um, but do I have specific recommendations? I don't. And one of the things that I think we need to be very careful about when we're looking at studies is this thing that says, well, people who exercise more do better. Um, but do they exercise more because they feel better? Like, or is the exercising improving them?
Right.
And I think we don't know that because some patients, they may not even have a big tumor, but it secretes some chemical that makes you feel like crap, and you don't exercise, and that chemical is also associated with progression. So, yes, of course, I believe in exercise, I believe in good nutrition, but I don't think we have done the right studies to figure this out.
Are you available to be contacted like after this presentation for referrals and so forth?
Yeah, of course. Yes, we're happy to.
Thank you. Yeah, thank you.
The one thing I will warn you because is I won't do email consults, but we're
Of
happy
course.
to see the patients and referrals. So I want to help patients, but email consults is not helpful to most
Can you
people.
provide contact information? And of course, I understand that.
Yeah, Brad
Right.
has my
Okay.
information. He can
Okay.
circulate
Thank you.
that. So we're happy to see patients. Um, other,
Ray,
yeah.
Ray, I can see already that we have 13 questions. Do you have a hard stop at the hour?
Let me just look what I have next. Just give me 1 second on my calendar. I have 15 minutes till
Okay.
my next meeting, so I can go a little over.
Okay, thank you. I have one that actually was triggered by Alan Morris. Implicit in the drug combinations and matching with multiple biomarkers is a fairly complex, I don't know, algorithm. And you mentioned AI can help or, will help and is being developed to help. Can you just sort of expand on that a little bit more, please? You just mentioned like on one of those patients, there was a MEK pathway plus an immunotherapy. It seems a little bit like I don't know, not alchemy, but like some expert decision making that you're making, but it's judgment calls. And over time, the models will get better, the data will get better. Can you just speak to how you see that evolving?
Right. So I think ultimately it has to be through companies that develop algorithms. And that's one of the reasons we founded CureMatch because when I was at UCSD, um, we would sit and Google what combination during molecular tumor board, what combinations should be done. And there was a supercomputer center, there is at UCSD. And so we worked with them and ultimately it grew into a company. Because a trillion combinations, and that actually underestimates the combinations because that's just based on genomics. And if you add in transcriptomics and immunomics, it goes beyond that. Um, is not something that anybody I know can do in their head. Um, so this really needs to be done at a level, computerized level. And I think ultimately this can only be done by companies that have the resources to put this together. And then I'm going to give you the example of Pangea, which we used for one of our patients, because it's actually a competing company, but that's fine because I like what they do. So they look at the transcriptome, at the RNA, and they look at something called synthetic lethality combinations, which decrease the viability, the ability of the cells to live. So they have a completely different algorithm, um, but I gave you an example of a patient who went from hospice to complete remission, and it was completely non-intuitive. We would have said this patient will never respond to immunotherapy. She had no known biomarkers. In fact, all her biomarkers were negative for immunotherapy. But at the RNA level, she did have biomarkers that their AI-based algorithm could come up with. So I think that there are going to be more than one way to look at this. Ultimately, will companies come together and put these algorithms together and we'll build on it? Absolutely. But the main thing that I'm trying to say is that the complexity to do the best job, the complexity is beyond human cognition alone. So I think I'm pretty good at this, and I know what I like and what combinations and what things I favor, and I can go through it and why I favor those things. But ultimately, you need assistance from a computer because, again, I don't think anybody can process a trillion combinations in their head.
Thank you. And we'll be talking to Bob Gattenby about that, where he works with these mathematical models and see how he does it in his work.
And Gattenby, I couldn't talk to him, but I think we have a new time. But he has a completely different theory, which is really interesting. I'd like to hear more about it. And so again, there may be more than one way to skin this cat.
Going back to the questions in the chat from Russ, when resources or tissue are limited, how should patients and clinicians prioritize among whole exome sequencing, RNA sequencing, proteomics, and liquid biopsy?
So liquid biopsy you can always get because it's a new blood sample. So that's easy. We do liquid biopsies on everybody, and that shows shed DNA from multiple sites. And so that's the advantage of it. The disadvantage is that it's probably not as sensitive as getting a piece of tissue. Um, our own prioritization is we do next generation sequencing. We do NGS as a first. Uh, and we use several different companies, commercial companies, to do NGS. Um, I can say not in any order that the ones that we find that are very reliable are Foundation Medicine, Caris, Tempus, Guardant, Boston Gene, um, Natera. Those are the ones that we use very commonly, and they have a spectrum of offerings. But we've been very happy with the information we get from any one of those companies. Um, so we prioritize liquid biopsy, everybody, NGS as first priority. We also have developed in-house a panel of 17 proteins that we look at by immunohistochemistry, which doesn't take up a lot of tissue. And if you asked me a decade ago, I would say that immunohistochemistry is not very valuable because targeting it is not that great. But that has changed now, in my opinion, because we have new, very powerful drugs like antibody drug conjugates, drugs like Enhertu, which is a HER2 conjugated to a chemotherapy payload. And so now we feel that immunohistochemistry and matching the abnormal protein with some of these antibody drug conjugates is really a powerful way to determine therapy as well.
Thank you. This is a specific to prostate cancer question. I don't know if you're conversant in that, but how can prostate subtype classification inform treatment selection and sequencing? These include luminal differentiated, luminal proliferative, basal immune, and basal neuroendocrine.
So, what I stated before, which I know that other physicians don't agree necessarily, is uh, I am oriented to the molecular classification and not to the histological classification. So, what I want to know is what are the molecular alterations in that patient? And to me, it kind of doesn't matter what the histological classification is because it's the molecular alterations that are driving the cancer. And it is true that some molecular alterations segregate um, with, to some extent, with histological considerations. but that's only helpful when you don't have access to NGS to say that in this histologic category, it's more common to have this abnormality. But once you have NGS, I don't need to guess what the abnormality is. I know what the abnormality is because I can test for it. And in addition, there are many tumors which I kind of say don't read the books. They have abnormalities in them that they shouldn't have. And yet, that's the abnormality. And I'll just give you one example is from the literature. Um, BCR-ABL is the hallmark of chronic myelogenous leukemia. We talked about that before. It's very targetable. The disease has been transformed from a uniformly fatal disease to one with a near normal life expectancy because it can be targeted. And BCR-ABL is absolutely specific for CML and for certain types of leukemia. Yet, in JCO Precision Oncology, probably about a year ago, maybe a year and a half ago, there was a publication of a brain tumor that had a BCR-ABL abnormality, and that patient responded to BCR-ABL inhibiting drug. I think it was imatinib. There's several other drugs in that class. So I kind of jokingly say that brain tumor didn't read the book. It's not supposed to have BCR-ABL. BCR-ABL is supposed to be for CML. Why the heck did that brain tumor have a BCR-ABL? Well, I don't know, but you would have never guessed that that brain tumor would have a BCR-ABL. But we don't need to guess. We just need to do the NGS. And so this is why, to me, the histologic classification is not what I'm looking for. I want to know what the NGS says.
And sort of building on that about question about RNA sequencing, does RNA sequencing provide a more sensitive method to detect early clonal escape mechanisms that may not yet be evident at the DNA level?
The answer is yes, but it's not that that's all it provides. It provides other things that are very important. And I'll give you a couple of examples. One is detection of fusions. BCR-ABL is a fusion, which happens to be an easy one to detect, but not all fusions are easy to detect. And some of them are very hard to detect at the DNA level. And RNA sequencing, in a recent study that we're about to submit to ASCO in rare tumors, RNA sequencing increased fusion detection by 33%. Now, why is this important? Fusions are critical drivers. We published a paper on this, and other people have seen that if you have a fusion, that is the driver, most likely. You need to target that fusion. So RNA sequencing helps with that. RNA sequencing also helps with other things. Sometimes you have a gene that you cannot target. We don't have a target for that gene. But you look at the RNA and you see certain RNAs are upregulated and downregulated. Um, and those can be targeted sometimes. And so we have examples of untargetable alterations. Uh, is, but then at the RNA level, we see things that are upregulated that we can target, probably as a result of those untargetable alterations. And we see patients that respond by targeting those RNA changes in expression. So I think RNA sequencing is important for multiple reasons. Those are two of them.
Another question here on next generation sequencing, and it might get to the question of whole exome, whole genome, but do all NGS tests use the same range or panel of tests? Wondering if some patients do not get the full panel but walk away with just a few biomarkers that have been identified.
So I mentioned several companies, and I'm going to mention them again, but uh, Tempus Caris, Foundation Medicine, Guardant, Boston Gene, all have very complete panels of, uh, let's say, 500 plus genes. So they are all very reliable and all, we would use any one of them. Natera has minimal residual disease, and they are going to come out with a big panel, I think, within a year. And they've been very uh, successful in the minimal residual disease space. Um, there are other panels, often they're local, institutional, that are smaller panels, 40 genes or 80 genes. I am not a fan of those panels. I think that they're too limited, and there's no reason not to do a full panel. It costs just as much to do 40 genes as to do 700 genes. You might as well do the 700 and have all the information.
The next question is staying with, I guess, this sort of testing. Related to AI-based approaches, there's one called ID tracks, which cell-based screening of small molecule compounds to their kinase inhibition data to rapidly personalize drug combinations. Have you heard of that? And do you have a comment?
I'm not familiar with ID tracks,
Okay.
but what it sounds like is it sounds like a kind of functional assay. In other words, trying to figure out, not by the gene abnormality, but by the actual function, what is aberrant. So let me tell you what I think is positive about these assays, and then let me tell you what I think is negative, because there's also assays where you grow cells or you grow organoids, and then you see like what combinations should we give to the patient. So the positive is that instead of algorithmically trying to figure out a trillion combinations, they're looking at the actual function in a cell-based system, and then we can take it into the patient. Um, the negative is um, if there's, I mentioned if there's 300 drugs in oncology, there's 4.5 million three drug combinations. None of these systems currently can test 4.5 million combinations. Maybe on a good day, they can test 100 combinations. So to me, the negative is how do you get to the 100 that you're going to test? You need some computerized algorithm to get to the 100, and then they can test it on the cell and see which of those 100 combinations. We're doing it mathematically. Let's say a CureMatch or a Pangea is doing it mathematically. Here, they would be doing it on the cell-based system, but they still need somehow to get from 4.5 million to 100. And then to pick between those 100, these functional systems might be lead us to the final path of those 100.
Okay. Can Canadian patients enroll in the WIN consortium for treatment?
Um, so, yes, so we do have a Canadian, um, the McGill, the Cancer Center at McGill is part of the WIN consortium. So we don't, the trial is not open, but we do have a molecular tumor board, and uh, the Montreal Center participates in that molecular tumor board.
Skipping ahead to one question related to that, how is your molecular tumor board membership chosen? We have an MD in our group, Alan Morris. He says, can any MD join this worldwide molecular tumor board, or do you have to be invited?
Um, you have to be invited. If somebody's interested, I chair the molecular tumor board. They can write to me. If the institution is a member of the WIN consortium, that helps. But we do have some world experts that their institution are not member, but they were still invited because of their expertise. Um, so I'm not the only decision member maker. Wafiq Eldiri is the who is the head of Brown University Cancer Center, and several other people make the decision if we are going to add people to the molecular tumor board. But if somebody's interested, they can write me and say I'm interested, and then we can go from there.
Okay. Just 2 more minutes.
Somebody said I don't read emails. I do
Uh,
read emails. I spend half my life reading emails. Um, so I do read emails. What I had said was that I won't do a patient consult, an email patient consult, because I think it's not helpful to the patient to consult by email. But what is my email? Brad Power can share my email address. I'm happy to share it.
A couple of specific cases here. I don't know if you're comfortable commenting, but we can maybe talk about them hypothetically. Here's someone with a rare ovarian cancer, 1% of endometrial cancers, done frontline and now facing recurrence potentially on the liver, 18 months after chemo and 14 to 15 months after Avastin. Surgeons are always saying that if after surgery I need chemo, they would use the same chemo, which is a combination of carbo and Taxol, which is what she had for frontline. Is this how your group would approach the recurrence, whether the lesion is on the liver or peritoneum?
So I don't know enough details about the patient to answer, but if you want to contact me, I have two ways that we might be able to help you. Um, or three ways actually. One is that we have a trial called the TRACK trial, which is run by the Target Cancer Foundation, which we mentioned earlier. And it is a decentralized trial across the United States. And if you have a rare cancer, you can enroll on the track trial. I can connect you. I'm the principal investigator, but we have investigators from 8 institutions across the country. I mentioned Harvard, MD Anderson, City of Hope, and so forth. And you will get a formal molecular tumor board consultation as part of the trial. The other possibilities are coming to our precision medicine clinic at Molecular College of Wisconsin. And the third possibility is depending on where you're located, there may be someplace closer where I'm familiar with people who practice the way we practice, and I can connect you, depending on where you live in the country.
Okay. In respect of your time,
Yeah,
Ray,
I actually have to get off.
yeah, let's wrap. What I'll do is I'll take the additional questions and send them to you, and maybe you can respond by email. In most cases I have email addresses. Charles, I don't have yours, for example. So if maybe if you're comfortable sending me an email, bradpower@cancerpatientlab.org, and then I can connect and we can ask your question and take these offline in email. Thanks, everyone.
Thank you. And